All right. Hi, everyone. Thank you guys for coming. So this will be our last week of SI this week. We have our session this evening. We're just going to go over practice questions, and then we have Thursday morning. Thursday morning. So the reason I'm doing practice questions tonight is because Bender's students took their lab exam today. So if you have any questions on the lab exam, I'll just really quick do a few minutes in case there's anybody in here from Knutson's class. Someone did write me a message asking if the muscle packet would be on the exam. So just in case there's anybody in here who is taking their lab exam tomorrow and didn't realize, your black and white illustrations will be on the lab exam. Okay. They will be in and all the numbers will be whited out. But if you label them, you're going to have the same ones that you practiced labeling will be on the lab exam. Okay. If you go to the guide to muscles, which is in your study resources module, that has all the answers to the black and white illustration. So just in case someone missed that and didn't realize, these will be on the lab exam. Okay. And again, you can go to your guide to muscles right here to find the answer key to the black and white illustrations. If you're in Bender's class, your packet should be due tonight, I believe. I believe it's due 24 hours, like within midnight of the lab exam. Any questions about the packet? Remember, you'd be doing origin, insertion, action, innervation for every muscle, handwriting it. If a muscle appears more than once, you can write on the second time. You can say, see page 1 for origin, insertion, action, innervation. Does that make sense? Like you already did it. You don't have to do it a second time for the same muscle. But for every new muscle, you need to do it again. There's a link at the end of Guide to Muscles you guys can follow here if you want like a link. I know I've already shown this to most of you, so that's why I'm kind of going fast, but it is here in case anybody missed that kind of info. The other thing on the lab exam is your muscles. You have your pink arm, red leg, torsos, the little torso, the big torsos. Good. And then remember, you have all your other models, sarcomere, the neuron. We went over all of this in SI at one point or another. Neuron, not loading, brain, spinal cord on the whiteboard and the woodboard, eye, ear, kidney, male reproductive, female reproductive. And then I already showed you the lab packet. Good. So in case anybody was in here that has not taken the lab exam, hopefully that's clear what you would be responsible for testing on. Okay. And then Bender students that are in here should have already taken it today. All right. Any lab questions for me, including if you guys were working on finishing your packet to submit it, anything like that? Remember, if you do not submit it, I think for Bender students, it should be by tonight, but maybe it's by tomorrow, but check your due date. But if you don't do the origin, insertion, action, innervation for each muscle, you will get like less than half the points. Okay.
Brooke, I have a question.
Yeah.
So in terms of, if you can give us kind of like broad percentage, like I know that the majority is going to be like the torso, the arms, and the legs. How much is going to be the other models, like the eye, the ear, and the male and female reproductive?
I don't remember my exam exactly. See, the bone exam was easy because it was like the front of the room. It was only the back row that I know had the models. But on this exam, I know they're like intermingled through the exam, if that makes sense.
Yeah.
So it's not like one row is models and one row is this. I know that Knutson said to me the last time I talked to him that the front row of the room is the illustrations from the packet. I don't remember perfectly well because, like I said, on my exam it was like mixed. I think he's remembering wrong because I remembered it would be like the first station was an illustration, the next one was a leg. The third station was a model, the next one was an illustration, the next one was an arm. So it was like very mixed up. Um, I would probably say I remember every model being on my exam. Like like not every single one of the kidney models was on my exam, but at least one of them was. Does that make sense?
Yeah.
There was the male on my exam. I had the male reproductive female. I had the ear and the eye. I don't remember if I had both spinal cords, but I had one with a lot of questions. I think I might have even had both. I know I had the brain. I can't remember if I had a neuron. I think I had a neuron. And then I definitely had the sarcomere. So
Okay.
all of them were on there at least once. And then you know that that's like minimum 2 or 3 questions on each one. I know there's 140 questions. So
And are they all multiple choice, right? For Newton?
yes, he said he changed them all to multiple choice now. So everything should be multiple choice.
Okay, thank you.
Yeah. Sorry, I can't give you exact there. I actually don't know the answer. I just know that I think all of them should be on there.
Okay. Well, because I just wanted to make sure because it's like you want to study everything just in case. I just
Yeah.
wanted to like hit every. I would rather study everything and go in knowing more than maybe I skipped a model or two.
Yes, exactly. Um, he told me 90 questions on muscles, half models, and half packet. Oh, I
Okay.
see. I see. 45 and 45. Yeah. I mean, I remember feeling like everything was tested. I remember walking away and being like, wow, I'm really glad I studied everything. I'll tell you honestly, the only thing, if I'm like really trying to give you a pointer, it is the only thing I can't remember if this was on the test, but
The neuron.
the neuron. But
Yeah.
I even will hesitate to tell you it's not because I know Nissl bodies are new this time, and I feel like I remember those being on the test. So I think this might have been on there too. So I think that it was all on there. I really do. I think all the models are on there.
Okay.
So I would definitely go over all of them. I know the muscle packet is on there a lot. In terms of if you want like my tip of the thing that I know was on there that's the hardest, I know that a lot of the pages, not every page will be on there. Okay, just we all know that not all the pages can be on there. There's 13 pages, but
Right.
the 2 hardest pages are both on there. Both of the arm are definitely on there. I think
Okay.
I think you're going to end up with like 10 of the 13 pages. But I know the neck and both of these arms are the 3 hardest pages in the packet. And I know they're all on the test. So just in case you were kind of like not studying one of those, make sure you study them.
Okay. My thing was like I made sure that I spent like multiple days on the muscle packet because I thought every single page of the muscle packet in the arm and the leg would be on there.
Yeah, I don't remember. It's 10. I would say 10 pages out of she just said 45 questions. So if
Right.
there's 45 questions out of this, I think that every page could potentially be on there. Just not every single muscle will be tested on every page. I'm going to be honest, like if I was creating the exam, if you look at this page right here, and then you look at this page, would you put both of those on there? They're like the same things. You
Yeah.
choose one of them, right? And
Right.
like, you're kind of going to look at like, do you know which one's going to be on the test? No, I'm just showing you that like, you're not going to have page 1 and page 2. It's just going to be page 1 or page 2, but you don't know which page. So study both of them. But you know
Right.
what I mean? Like, so not all of these can be on there because it's like, some of them are kind of the same thing twice. So that's like a waste of your time.
Okay. All right.
Good question. No, good question. I'm glad we clarified. Okay, any other questions for the lab exam? So I don't know what else to tell you other than know everything. It's all going to be on there. But 140 questions, it will be worth your effort to go over everything. It's not, a disappointing few questions. Okay, so my goal here is to transition over to lecture. I don't know what else I would be going over for lab. unless anybody has any questions. So if you have your lab exam tomorrow, I would probably recommend focusing on that. So if you have your lab exam tomorrow, unless you have other questions, maybe, touch on the recording or something, I would probably recommend that you guys focus on your lab exam tonight versus necessarily being here to listen to practice questions. Again, would be if you have your lab exam tomorrow. But if you feel really prepared, maybe practice questions are more helpful. To be clear, where you guys have your practice questions. Study resources. I probably said this 12,000 times. Study resources. And then you should have like over 100 questions. I'm thinking it's over 150, maybe close to 200 questions if you click on each of these. Does that make sense? All of those are links. These are the ones that I made the most recently. The most recent. I think this is 50. I think I tried to make 50. Yeah, I made 50. So this is what I plan on going over. They're already posted, but I do feel like it's helpful when we kind of go over the questions because then it's kind of more like review. Okay, so let's start with this. This is just to list the topic. So this is muscle. Okay, I'll see you guys out there and then we'll start.
Recording in progress.
Okay, so remember, extensible is stretch. So the answer here would be D, extensible. Elastic is the ability to return to its original shape. So, when you have like I don't know, you probably can see my camera somewhere in there. If you have a hair elastic, right, and you stretch it, that's extensibility, extensible. But when I let it go and it returns back to its original shape, that's its elastic nature. Does that make sense? Extensible, the stretch. Elastic, when it returns to its original shape. Good? So our answer to 2 would be D, extensible. Extensible is stretch. Elastic is return to original shape. 3, muscles do not do which of these? Okay, so I would also say D. D is kind of tempting because it sounds a little weird, help move minerals, but they actually do help with the movement and the storage of materials and different substances. So I think that we're going with E because all are correct. Make sure I wasn't on some other thought. No, I did go with D because I have materials. That's kind of a tricky question. I don't know. Probably a little too complicated. So technically, they help move materials, not minerals. Okay. So D is the correct answer because they help move materials. Materials are examples are calcium and glucose, which are not minerals. Minerals are like, you know, boron, I think is a mineral, and like hydrogen or something. So, you know, technically they're helping move other materials that are not minerals. Does that make sense? I'm probably going to copy and paste that into our answer key. Okay. 4, which of these is not true of myofilaments? Okay, so if we choose E, you guys are saying A, B, C, and D are all true. So let's look at each of them. A, actin is a thin filament. We know that that's accurate, right? So let's draw, I'll draw a little actin. You know, here's my actin. It kind of looks like this along the cell, right? That's my light band. And then you have your myosin, which is the thick filament, right? Or the dark band. I'm on my computer, so I know my drawing here isn't that great. So myosin is a thick filament. So A and B are both true, so those can't be the right answer. And they do not have a connective tissue covering, but actually C is the correct answer because this is not true. So if you look, if this is my muscle cell, my myofiber, right? Sorry, I know that got a little wonky there. Okay, this is my myofiber. Picture, let's go back to this right here. Okay, let's look at this really quick because I feel like it's probably even better than a drawing, right? So look here. This is my myosin, my actin, my myosin, and my actin. Do you guys see how they do not run the whole length of the myofiber? Instead, they break up down the myofiber. Does that make sense? They're like little sections that break up. So when it comes to the cell, the myofiber, these are myofibrils. So I'm going to do like a little, just real quick. Where's my thing? I've lost my annotate. Come on here. Okay. I don't know why my annotate is gone. There we go. Okay. So you have your muscle, which obviously runs the whole length of the muscle. Fascicles run entire length of the muscle, right? The myofiber is the next one. Those run the entire length of the muscle, meaning your whole muscle length, right? That's going down it. And then your next division is the myofibril. And these run the entire length, right? And then making up each myofibril is the myofilament, which is our actin and myosin. And these are the only ones that do not run the entire length of the muscle cell. You're very likely to see that on the exam. Okay, you're probably going to see a question on that. You know, there's 165 questions. So that's a topic. This is a pretty key point here. So you're likely to see that. Each of these would be a myofibril. Okay, each of those round things. And then as you look down here, do you guys see how this would be the myosin? This is the actin, myosin, and actin. Do you guys see how those are breaking it up? Those are not running the entire length of the myofiber or myofibril, right? And this entire thing would be our myofiber. Good? Hopefully the visual was a little bit helpful for that. Okay, let's go back here. Okay, let's move on to question 5. The superficial fascia help to separate muscle from muscle. Okay, this might be an area where we haven't looked at this. So superficial fascia, this is going to be the most superficial. So if you think about the most superficial, what is the most superficial thing you can think of? Your skin, right? Your skin is the most superficial. So this separates muscle from skin. Maybe think superficial S for skin, okay? Deep fascia separates muscle from muscle, okay? So kind of think of it, they're deeper down, right? And then our next three are going to be our epimysium. This surrounds the entire muscle, and this is made of dense, irregular connective tissue. I know I gave away the next answers, but I feel like this is probably good to put here, okay? Perimysium, this surrounds each fascicle, okay? And this is made of dense, irregular connective tissue. And then lastly, our endomysium around each myofiber. And this is made of loose areolar connective tissue. Okay, so those are all your coverings. I would, again, with 170 questions, you kind of want to make sure you're touching on everything. This is pretty easy to ask you questions on. So obviously, question 6, okay, question 6, is this true or false? So let's just go through them just to make sure you guys get it. 6, true or false? The deep fascia surrounds the muscle to protect it. So the deep fascia separates muscles from each other, right? What is actually surrounding the muscle itself is the epimysium. Does that make sense? So the answer to 6 would be false. The correct answer would be epimysium. Does that make sense? The deep fascia separates muscles from other muscles versus epimysium protects the actual muscle. Okay. Question 7, dense irregular connective tissue is found where out of these options? This got very tricky. The wording is very tricky, and that's not what I meant. I didn't mean around. Just cross out around the and just say perimysium, endomysium, myofibril, or myofilaments. So I think I was going, what am I trying to, I don't really see what this question is trying to say. The correct answer here should be, should be A, because A is the only one that is made of dense irregular. Let me see what I put. I might have been, my train of thought there isn't clear. 7A, yes, endomysium has loose areolar, all the others don't have connective tissue. So sorry for the weird wording there. It shouldn't say around the, that wasn't what I meant. So again, loose areolar is endomysium, perimysium has dense irregular. Does that make sense? And so does epimysium. The myofibril and myofilaments don't have any connective tissue at all. So 7 really is a bad question. Question 8, which is not true of the myofibril? Okay, so the myofibril. So kind of refresh your memory. What is the order? And then we'll answer it. It is helpful for me when you guys write your answer in the chat. If you don't want everybody to see your answer, you can send me a direct message. I'm really just looking so that I can see if you guys are done answering the question, and then also if you need kind of clarification. If you guys answer the wrong thing, it helps me to know if there's a way that I can kind of clarify, but I won't say who said anything. If you want to send it direct to me, that's fine, or to everyone, but then I know if you answered it. So for number 8, we'll just go ahead and answer that, and then we'll try for the next one for you guys to put it in the chat. So for number 8, the D would be the answer because the myofibrils and the myofilaments have no covering. So if you look on the left-hand column, we talked about muscle from skin, muscle from muscle, the muscle itself, the fascicle, the myofiber, and then you know that there are 2 more categories, right? After the myofiber, we go to the myofibril. This has no connective tissue covering, and then the myofilament. The myofilament has no connective tissue covering. Myofilament is actin and myosin. Does that make sense? So our answer to number 8 would be D, because it does not have dense irregular connective tissue covering or any covering, so that's not true. Okay, question number 9. Which of these marks the outer limit of the sarcomere? Good, it's the Z line, right? Z line can also be called Z disc. They are just names for the same thing. So it's Z line or Z disc. Definition, it marks the outer limit of the sarcomere. Question 10, which of these contains the full length of myosin? Right, so remember that the full length of myosin is part of the A band. That's the dark band, the A band that has the full length of myosin, okay? Actin is in the I band, and it's a little bit in the A band. So what happens with the sliding filament mechanism is that the I band basically moves towards the center and the H zone disappears. So the 2 I bands which have actin, think actin has the letter I in it, right? Actin, and it does the action. Actin does the action and it moves towards the center. So there's a little bit of actin in the A band and in the I band, but the A band has myosin only in terms of, you know, across the full length of it, versus the I band has like actin and some titin, but there's also actin in the A band. So if the question were to say, which of these contains the full length of actin? Okay, so if the question, let's do this. If the question were to say, which of these contains the full length of actin, the full length. So the full length means all of actin. So this would be all. Which answer would you choose? So let's re-answer. Which answer would you choose if it was, which of these contains all of actin? Okay, so you're thinking D, but that would not be accurate, right? So the answer would be E, okay, because the I band does not contain all of actin. So actin is found in both the I band and the A band. Okay, so remembering our little drawing, this was our little dark band, right? Here's our, let's do it in green. Here's our light band over here, light band. When contraction happens, these light bands move towards the center. The area here without any actin is called the H zone. So when contraction happens, the H zone disappears because the actin moves to the center. Now, if you guys look, right, here's my actin here, actin. And you guys see how the actin is partially inside the A band zone and partially over here. So the I band is right here, I band, and this is the A band. The I band is actin, yes, but some actin is also in the A band. That's why there's an H zone. So on your test, the definition of the H zone is the absence of actin, okay? So remember, actin is found in the I band and a little bit in the A band. That's what creates the H zone, meaning the H zone is the part where there's no actin at all, right down the middle. Does that make sense? When the actin moves towards the center, right, the H zone or the absence of actin disappears. The I band is not the full length of actin because some actin is in the A band. Does that make it more clear? The myosin is the full length of the A band, right? The A band has myosin along the full length because this red that I drew here, I should have done it in red, but this red guy right here, that would be myosin. Hopefully that helps. It's a recap. If you guys feel like you're really confused, maybe write down on a piece of paper, okay, I need to go over muscle contraction one more time because I'm not really getting any of what she just said. So maybe you need to check out, I'm going to remind you, if you go in this YouTube videos and helpful links in the study resources, there is a video in here that I just love for muscle contraction. It really helped me when I was in the class. You can kind of scroll down because this will all be like unit 3 videos at the bottom. Right here, this one, Muscles Part 1, Muscle Cells Crash Course. I really like this. I think it's like, yeah, it's 10 minutes long. That's worth your time. It's very comprehensive, but towards the end of the video, he kind of does a whole diagram where he shows you how muscle contraction works. And then he also, I think, goes over it on the microscope somewhere in here. Anyway, so I would really recommend watching this. I think it's really, really helpful, and it'll kind of give you a visual. You can also look at what other videos are in here. Okay, so hopefully that inspires you or helped a little bit regardless. Okay, let's look at 11. Which of these helps hold the actin in place? Okay, so you're picturing that microscope drawing. Which of these helped hold the actin in place? So our answer here would be E. Titin helps to hold the myosin in place. So nothing holds the actin in place. Titin helps to hold myosin in place. That was kind of a tricky one, but our answer there would be E or none because titin helps hold myosin in place. Okay, question 12. True or false? The H zone is defined as the absence of actin. True or false? True, right? We just went over that. Perfect. Okay, question 13. Troponin attaches to so you're kind of moving on to muscle contraction inside of the cell, right? Good, good. Calcium ions, right? So you remember that, you know, we have our little, I'll kind of just draw the outside. You have your little tropomyosin, and then troponin is kind of attached, like little snake things. And our calcium binds to that troponin right here. Okay, and we know that that will bend the tropomyosin when that happens. Okay, question 14. The active site on actin. Which of these is not true? Good. So we know it would be A, right? Actin is a part of the I band. So the I band has actin. Remember we said that actin is in both the I band and the A band, but it is also in the I band. So make sure you know actin is in the I band. So the actin is a part of the I band, but it is not covered by troponin. It's covered by tropomyosin, right? So if we draw our little actin with its active site, these would be little balls of actin with their active sites, and they are covered by tropomyosin, not troponin, but tropomyosin. These little squiggly guys, those are the troponin. Does that make sense? Troponin attaches to calcium. Tropomyosin covers actin. Okay. And then we know that our little myosin heads wait up here to form a cross bridge, which is contraction, with that active site on actin when we bend it away. And remember that our starting point for all of this is when we have that T tubule and we get what? Sodium comes down the T tubule. And when sodium comes down, it triggers the sarcoplasmic reticulum to release calcium out of the terminal cisterna, okay? And our calcium, you know, we have our little calcium molecules, and those release out of the terminal cisterna, and they go and they attach to the troponin to bend away tropomyosin, right? So our sarcoplasmic reticulum and terminal cisterna. Okay, so question number 15. Let me scroll down because we can't see the whole question. Okay. Question 15. A single muscle contraction and relaxation is called a what? B, right? A twitch. Good. 15, B is correct, twitch. 16, true or false? Contraction without relaxation is called a twitch. False, right? We just said that. So what is the correct answer for 16? What could we rename this and make it a true statement? There's two answers that we could rename this. So why don't you go back here? Tell me which two of these are correct for number 16. So you're going to choose two here. Well, not E. Two of these are correct. So it's A, tetanus, or D, cramp. A and D. A and D. Tetanus refers to contraction without relaxation. Tetanus occurs, I have my caps lock on. Tetanus occurs when the muscle loses ATP. So remember, ATP is required to undo the muscle contraction. So if you have no ATP, you have no relaxation. And this occurs in tetanus. I believe tetanus occurs when like toxins, I don't really know much about tetanus, honestly, but I think tetanus has to do with toxins blocking the receptor sites to make it relax, but no ATP. So tetanus or cramp. A cramp happens to all of us, right? We all get cramps every now and then in our muscle, but tetanus is a condition when somebody's whole body is contracted without relaxing. But for the sake of your kind of knowledge for this class, contraction without relaxation can be called tetanus or cramp. I'm not really sure why it's not called a spasm, but I don't have spasm in my notes, so we're not going to go with spasm. Okay, because spasm would be just us making up something. I'm not really sure what they call a spasm. Maybe like a seizing of the muscle that goes away. Okay, so got that? Tetanus or cramp is contraction without relaxation. Okay, question 17. The neuromuscular junction does not involve which of these? Yeah, Nora, what's up? What's your question?
Okay, so I was just going over the sarcomere and I realized that there's something called a motor end plate. Is that connecting, like it's like the squiggly part that starts on the top and it goes down the side, but is that also the sarcolemma?
You know, that's a great question, and we're actually doing that question now. So let's all go over there and then we can all kind of see it. So if you guys were answering that over here, the answer to this is C, because the neuromuscular junction involves the synaptic knob communicating with the motor end plate on the myofiber. So if we go over here, the question she's asking goes with that question, because this motor end plate is this little squiggly part right here. And then this yellow is the synaptic knob with the little, see if I can get it to zoom, with the little synaptic vesicles inside that have the neurotransmitter. This is the synaptic cleft, then the motor end plate. So what you see here is a neuromuscular junction. You're asking, is the motor end plate the same as the sarcolemma? Yes and no. The portion of the sarcolemma that is wavy is the same as the motor end plate. But not all of the sarcolemma is wavy in order to accommodate the T tubules. So the motor end plate is a part of the sarcolemma, but they're not the same thing, because the part that's wavy is the motor end plate. Does that kind of make sense?
Yeah. What is the other name you said? Cleft? Is that the part where it's straight?
That would be the blue where there's a space. Oh. Synaptic cleft is the space in between the two.
Okay.
Because this is synaptic knob with synaptic vesicles. On your lab exam, if it was pointing to the squiggly area, you would want to say motor end plate. If it's pointing to, you know, this straight area or the smooth, usually we would label it right here. That's where we would be wanting sarcolemma. Okay, but that's technically motor end plate is the wavy part of the sarcolemma.
Okay. All right, thanks.
Yeah, no problem. Okay, everybody got that? So question 17, it does not involve a dendrite because remember the neuromuscular junction involves the motor end plate. A dendrite would be for a neuro-neuro junction. Make sense? Synaptic knob to dendrite, neuro-neuro. Synaptic knob to motor end plate, neuromuscular. Question 18, the presynaptic membrane is the same as which of these? So which of these is the presynaptic membrane? So that would be the synaptic knob, right? Because the presynaptic membrane is always the synaptic knob. Whether you are in neuro-neuro, here's my presynaptic in neuro-neuro, it goes to a dendrite, D for dendrite. In neuromuscular, it goes to the motor end plate. Does that make sense? Neuromuscular to the motor end plate, neuro-neuro to the dendrite. But either way, the presynaptic membrane is always the synaptic knob. Good? Okay, 19. The most common neurotransmitter is, which one? Some of the people that are leaving are going to study for Knudsen's lab exam tomorrow. E, none, right? So the most common neurotransmitter is acetylcholine. So none of those options would be correct. Option that's correct is acetylcholine. Okay. The enzyme responsible for ending a neuromuscular synapse is which of these enzymes? Good. So this one's just kind of a trick question. The A, B, and C are not ever something you guys have learned about. They're just to kind of throw you guys off. The right answer here is D, acetylcholinesterase is the enzyme that breaks up acetylcholine. So if you think about it, if you have all these acetylcholine molecules over here in that synaptic cleft, you guys can picture how they're constantly sending the signal. Does that make sense? But what we want to do is break up or digest all of those acetylcholine molecules so we stop the signal from carrying on. So to end the neuromuscular synapse, which is like ending the command, you would be using acetylcholinesterase. Okay, so that makes sure that there's no more signal kind of coming through. Question 21, which of these is not true of calcium? Good. B, right? So it's kind of like a trick question. This is the one that's the answer because it binds to troponin is the correct answer to bend away tropomyosin, right? So that's why we choose B because our wording here is wrong. It does not bind to tropomyosin to bend away troponin. It bends to troponin to bend away tropomyosin. Okay, so that's kind of like the reverse. All the others would be true, right? It comes into that axon terminal here. It breaks up those little synaptic vesicles that are inside. That's what calcium does. Calcium comes in here. Calcium breaks up those little synaptic vesicles. It also, when the sodium comes down, it's released from that sarcoplasmic reticulum here. That's where our calcium is released. And like we talked about up here, it binds to that troponin. Okay, question number 22. A motor unit is a single motor neuron and the single muscle fiber that it controls. True or false? So our answer here is false because it is all the muscle fibers it controls. Make sense? So not just one. So any muscle fiber that's controlled by that motor neuron is called a motor unit. Question 23. The prime mover is which of these? Which is the prime mover when we talk about muscle contraction? Good. A or agonist is the same as the prime mover. 24, the one that assists the agonist. Good. The synergist, right? And then 25, the smooth muscle cells do not have So remember, they do not have autorhythmia. Autorhythmia is not the same as involuntary control. Autorhythmia refers to the fact that, like, if you cut out someone's small intestine, it would not keep going. Does that make sense? But if you cut out someone's heart, the heart is able to beat without the nervous system controlling it. Without the brain's input, the heart can beat. The only cells that are autorhythmic are cardiac muscle cells for the heart. So the smooth muscle cells are not autorhythmic. That's kind of a tricky one because it's just supposed to throw you off. All right, question 26. We're moving on to spinal cord. So kind of transition your thinking over to spinal cord. The interneurons, which of these is not true about the interneurons? So our answer here would be B, interneurons are also called association neurons. So A is accurate. They do facilitate communication, and they are, you know what? I see what you're saying. Someone said that D isn't right. That is true, because they would be a neuron according to, well, no, I have them in my notes as a neuron according to function. Let me make sure. That's a good, I want to make sure I'm saying it right. No, they are a neuron according to function. It's kind of weird because they seem like they should be a neuron by structure, but the neurons by structure are unipolar, bipolar, and multipolar versus the neurons by function. Structure would be unipolar, bipolar, multipolar. Function categories would be motor, efferent, sensory, afferent, and interneuron association neuron. So D is a true statement. So the answer to this question would be B. They are not found in the anterior horn. Okay. So by structure, we have unipolar. Remember, unipolar neurons are found in the dorsal root ganglia. They are sensory or afferent. Bipolar neurons are for the eye, the ear, and the nose. Eye, ear, nose. I'm just kind of adding this little detail in here for you guys. They would be part of the sensory system. So we're going to put a little arrow there. And then multipolar neurons are the motor or efferent neurons. Interneurons are really kind of, they don't really belong in the unipolar, bipolar, or multipolar. They are like they're like an anaxonic multipolar because they have a lot of, we'll just do this. They have a lot of things and they don't have an axon. But this isn't really something you need to know for the test. You want to know for the test that multipolar are motor, bipolar are eye, ear, and nose, which would be sensory. And then the unipolar, obviously the dorsal root ganglion, and those are sensory. And then know that the interneuron or association neurons facilitate communication. They're a neuron according to function. They're also called association. They are found in the lateral horn. Lateral and a little bit in the posterior. Should be lateral and I think I have a little bit in the posterior as well. Yes. This is just me not wanting to accidentally tell you posterior and then not posterior, but I think we can say posterior. Where's my thing for that? Interneuron, lateral horns. Yes. Yes, so they're in the lateral and posterior for the location of the interneuron. Good. Okay, so moving on to question number 27. Question 27 here. Somatic sensory information. Which of these is not true about somatic sensory information? Let me get my thing to scroll down. There we go. Okay. Any guesses here on 27? Which of these is not true? So our little recap of our spinal cord, we have our back side posterior, and then we have our front side, and that is anterior. The information joins up together. So our front side is motor efferent. Our back side is sensory afferent. If it's somatic sensory, it has its cell bodies here in the dorsal root ganglion, and they end or they synapse in the posterior horn. Sorry, this little thing got funny looking. Okay, so posterior horn has the axon of the somatic sensory cell bodies. So our somatic sensory cell body, somatic sensory cell bodies are in the dorsal root ganglion, and they are unipolar. They're a unipolar structure, and they are maintained by satellite cells. So those would all be facts you would want to know for your test. Somatic sensory cell bodies are in the dorsal root ganglion. They're unipolar by structure. They're maintained by satellite cells. The posterior horn does not have any somatic sensory cell bodies. It's just the axon. So the correct answer to 27 would be C, because they do not have the cell bodies in the posterior horn. Good? When we talk about motor information, remember motor cell bodies would be in the anterior horn for somatic motor, and they come out here. Somatic motor cell bodies are multipolar, and their cell body is in the anterior horn. The lateral horn has the autonomic cell bodies for motor and sensory autonomic. Remember, you have autonomic, sensory, and motor, and that's going to be over here. Let's do like a little different color, maybe blue. So here's my motor in my lateral and my posterior horn, and it's helping with my autonomic motor and my autonomic sensory that's coming in. Okay, so autonomic is involved in our lateral horn mostly, versus our anterior would be somatic motor, what you're aware of. Somatic sensory is the dorsal root ganglion. Okay, question 28. Somatic motor information does not Anyone else want to guess? Just have one guess so far. Okay, so our answer here would be C, okay, because somatic motor information is that green one I drew here. It does flow through the trunk of the spinal nerve. This section right here is the trunk of the spinal nerve. So the trunk of the spinal nerve actually has all types of information. So it would have somatic sensory, visceral or autonomic sensory, somatic motor, and visceral or autonomic motor. So it's all there in the trunk of the spinal nerve. Just think of it as a nerve. The anterior root is the part right here would be the anterior root. Here is the anterior root, and that does have our somatic and visceral motor information. And then we know that its cell bodies are in the anterior horn, which I just showed you guys. It does not synapse in the lateral horn. So the lateral horn is for autonomic. So that's not involved with the synapse of the somatic motor. I know it's kind of a lot of terminology to keep track of, but you know, it's kind of worth reviewing. I would just say if you're kind of getting most of what I'm saying here on the right, and that diagram is making sense to you, and as we're reviewing it, it's ringing a lot of bells. The basics is what you should really be focusing on for spinal cord. if I'm telling you an encouragement of which one to go deeper with muscle contraction and all of that versus getting caught in the weeds with spinal cord. So if you're wanting advice of which one to study more, spinal cord will be on the test. Just to be very clear, it will be on your lecture final. Spinal cord will be, but not probably to the same level of detail as the muscle contraction. Okay, so which one to focus in more detail? Muscle. What should you know about spinal cord? Everything that I've already talked about would be stuff you'd want to know. Okay, so when I say not a lot of detail, it might be different than your definition of not a lot of detail. What I have written on the screen is my definition of not a lot of detail. Does that make sense? You still want to know everything we've talked about. But maybe you don't need to go into as much detail as, you know, getting into like, what is the white commissure and what are the funiculi? So there's a lot more stuff you can go into that you don't necessarily need to. So hopefully that helped someone and didn't confuse you guys. When in doubt, study more, don't study less, but study more on the muscle contraction because that's kind of where people get confused. Okay, question 29. Visceral sensory information, which is true. So you want to know these questions that we're going over. This would be stuff that you would want to know. Visceral sensory information, which is true. Good. That's right. So it is also called afferent, right? All sensory information is called afferent. We know that sensory information does not synapse in the anterior horn. It would synapse in the posterior horn. It does not have cell bodies in the anterior horn. That's motor only. And it does not come in through the anterior side. That would be motor only. So sensory comes in posterior, synapse is posterior, and it's also called afferent. All right, question 30. Autonomic motor, not true. Which of these is not true of autonomic motor? So remember, our autonomic motor is divided into two systems, sympathetic and parasympathetic. So we know that it's A is accurate. It is part of the autonomic motor system. B should be accurate. The answer here should be E. I'm going to go make sure that the answer is listed as E. 30E. Yes. So the answer is E because all of these are true. So the autonomic motor does supply the sympathetic chain of ganglia. The preganglionic cell bodies are in the lateral horn. Remember, this is for autonomic motor, not for somatic motor. Somatic motor has them in the anterior horn for motor. But autonomic motor, they're in the lateral horn. And then the postganglionic cell bodies, which are after the synapse, are always in the peripheral nervous system. Remember, the peripheral nervous system is once you leave the spinal cord central. And then we know that motor is always called efferent, whether it's autonomic or somatic. Okay, so that was kind of a tricky one, but all of those would be true. Okay, so this is kind of now into the brain, not in the spinal cord anymore, so kind of transitioning into the brain. Question 31, the thalamus, which of these is true about the thalamus? Any guesses? Thalamus. Okay, so we know that the thalamus is not located in the brainstem, right? The brainstem has the medulla oblongata, it has the pons, and it has the midbrain. So that's the brainstem. So we're gonna take that off of where the thalamus is. The thalamus is part of the diencephalon, right? The diencephalon has the epithalamus, thalamus, hypothalamus. Diencephalon, you can just think of hormones. You know, it's a really easy way to maybe remember diencephalon. Think of hormones, and then H, hypothalamus, has hormones. So it's the one with all the thalamus in it. Diencephalon has all the thalamus guys in there, versus brainstem has the midbrain, pons, and medulla oblongata. The pyramids are part of the medulla oblongata, so B would not be true. C is the function of the thalamus. So the thalamus sends sensory information to the correct spot on the postcentral gyrus, which is the same as the primary somatosensory cortex, with the exception of olfactory. So no smell goes through the thalamus. The thalamus does not do smell. So D would be wrong. It doesn't use smell. It does send information, specifically sensory information, to the correct spot on the postcentral gyrus. So C would be true. Okay. The epithalamus. So this is just kind of going over the functions within the brain. The epithalamus, what is true? Good. So the epithalamus has the pineal gland. The pineal gland releases melatonin. Good. So epithalamus has pineal gland, which releases melatonin. So the epithalamus does not release melatonin, but it has a pineal gland that does. And then the thalamus is the switching station for sensory information, sending it where it needs to go on the postcentral gyrus, which is the same as the primary somatosensory cortex, except olfaction. So olfaction or smell is not one of the things that is switched. All right, let's answer 33. The hypothalamus, which of these is not true about the hypothalamus? Okay, so B would be the correct answer because the hypothalamus does produce hormones, but it doesn't actually release them. It should be the pituitary gland that is releasing them. Okay, so the pituitary gland is the one doing the releasing of the hormones. The hypothalamus produces them and stores them in the pituitary gland. It's connected to the pituitary gland through the infundibulum, which is like a little tube. And the hypothalamus is found in the diencephalon. So our correct answer there would be B, because it does not release the hormones, it just stores them. All right, question 34. The cerebral aqueduct is found in, remember the cerebral aqueduct connects the third and the fourth ventricle. Good. So you guys probably know this because you've gone over the model, right? It's found in the midbrain. Good. So B, midbrain. I know these have been kind of harder questions, but hopefully you guys start getting it. We're going to move on to kidney next. Okay, question 35. The pyramids, which of these is not true about the pyramids? Good. So the pyramids are found in the medulla oblongata. Pyramids, medulla oblongata. And they switch white matter tracks. They perform what's called decussation, contralateralization, or crossing over. They're kind of like they kind of help just the white matter tracks, which is like the axons, switch over to the opposite side. They basically help the left side of the brain control the right side of the body, the right side of the brain control the left side of the body. You know, one of the ways you could remember brainstem and what's inside brainstem, look at the M and maybe just think for M, you could think medulla and midbrain. And for B, you could think B for breathing, because the pons, main function of the pons is breathing. So you could maybe remember it like that. Brainstem, medulla, midbrain, M, breathing for pons. Versus diencephalon is going to be your 3 thalamus, right? Thalamus, 1, 2, and 3. Epithalamus, hypothalamus, and then hypothalamus. Epithalamus, and then hypothalamus, right? Diencephalon. I don't know if I can really see a way to remember that, but maybe remember brain stem, M for medulla, and B for breathing for pons. Okay, so let's move on to question 36. We're moving into the kidneys now. Okay, so kidneys. Question 36. Which of these is true about the cortex of the kidneys? Good, C, right? It is the outer layer of the kidney. So you might have chosen B because there's the corticonephrons, but remember the corticonephrons do not make up the cortex. They're just the cell body, not cell body, the glomerulus of them is found in the cortex, but it's not what makes the cortex. The cortex is more like an outer layer that has a little bit of the corticonephron, but that's not what makes it up. The outer layer of the kidney would be the most accurate statement there. What is made out of nephrons are the pyramids. So the pyramids are made out of nephrons. All right, 37. The internal urethral sphincter is controlled by So if it's not, I saw your answer, Andrew. If it's not B or D, remember that the somatic system is going to be the same as skeletal. So if you've eliminated B, you've also eliminated A, right? So what do you think the answer might be then? It's going to be C or E, C. right? So we know that the skeletal or somatic system, skeletal muscle, is the same as somatic. There's also somatic sensory, which would be like what you are aware of sensing, just to make sure you guys know there's two somatics. But somatic system would be skeletal muscle. This is for the external urethral sphincter. So when you go to the bathroom and you choose to go pee, you're using your external urethral sphincter. Does that make sense? You're using your skeletal muscle, you're choosing to go pee. But when you feel the urge to go pee, that's your internal urethral sphincter governed by your autonomic nervous system. This is why when you're like stressed out, you know, if you were like, I don't know, in like a flight or flight terror situation, you don't feel like you need to pee at that moment. Does that make sense? So you pee when you're more relaxed, when your autonomic nervous system is telling you, okay, it's time, you know, it's time to go pee. Then you go to the bathroom and you choose your external skeletal muscle one to actually make the choice to go. Does that make sense? Internal, governed by autonomic, you're not aware, but then you feel like you need to go. External is what you choose to do, somatic or skeletal. Obviously, cardiac, no, because that's just the heart. It's not what's involved there. Okay, question 38. Which of these is not true of the nephron? Good. B, right? The nephron is made of just two parts. Two parts. It's made of the renal tubule and the glomerulus. I think it's the Bowman's capsule. Okay. The collecting duct is not a part of the nephron. That's like separate from the nephron. Okay, question 39. The fenestrated capillaries. Which of these is not true about the fenestrated capillaries? Are they part of the glomerulus? Are they afferent and efferent arterioles? Allow filtrate to pass out, allow water to pass out. It's kind of an odd question. I don't know exactly. It's kind of a weird one. Yeah. So I was going for B, afferent and efferent arterioles is the fenestrated capillaries. Technically, as far as you guys learn in this class, they don't allow water to pass out. I kind of forgot. I mixed up my classes here because in physiology, technically you learn that they do allow water. But for this class, we say only the loop of Henle allows water. So this is not like my best question here, but 39, B is definitely not true. So we can just go with that. B is definitely not true. So B has to be the answer. Does that make sense to everybody? D technically is also not true. So there's like two that aren't true. But for this question, we're just going to say B because the afferent and the efferent arterioles absolutely do not, they're not fenestrated, right? It has to get into the glomerulus to become fenestrated. So kind of sorry about that there, the D not being right. Okay, 40, the nephrons empty into which of these first? So which is the first place that the nephrons empty? A, right? We get to the collecting duct first. Renal tubule flows into the collecting duct. Okay, question 41. The renal calyxes empty into which of these? Good, the renal pelvis, right? So we have this up here would be the correct flow. You go from the nephron to the collecting duct to minor calyx is joined to the major calyx joins to the renal pelvis, which then goes to the ureter and out to the bladder, and then out the urethra. So that's the whole journey of the urine. Okay, 42. The ascending limb of the loop of Henle is permeable to which of these? So take a second and kind of think about this. Ascending limb. Okay, so I'm going to do a quick little recap, and then I'll give you like a little easy way to remember it. So we have our loop of Henle. This would be descending, and this would be ascending, right? So we have our blood that flows here. This is our vasa recta. I'll kind of draw it a little squiggly so we can see it. And it flows in the opposite direction, the blood. So what's going to happen is salt is going to leave the ascending limb and get into the blood. So this is salt, S, salt. So salt leaves the ascending limb. So the ascending limb is permeable to B, salt. And then what happens after that is water is motivated to join the salt out of the descending limb. So the descending limb is permeable to water. Why is this? Usually, I felt very confused when I was in the class. I felt like I didn't really get it. It's because the blood comes here first. Does that make sense? The blood starts here and then works its way around. So you have to think of the fact that the blood gets to the ascending limb first and then the descending limb. Does that make sense? So the way you can kind of remember this, it's like a little cheat thing. Ascending has an A, S, salt. Okay, I know that it's like kind of backwards, but you could think A for salt, right? Or you could also remember it, make sure you don't get confused. AS, both of those are in salt, like SA, salt, AS, salt. So that's kind of how you could remember that little, descending for water. I don't know if I can really think of a way to remember that, but maybe just think water, D comes in the alphabet after water follows salt. So descending will follow the salt. D, I don't know, I can't quite think of anything. I mean, you could think of water. Usually when we say it as Americans, it sounds like we're saying the D, water, not water, water. But anyway, if you can just remember A for salt, then you'll remember the other side. Okay, 43. Which of these is not true of angiotensinogen? So up here we did our loop of Henle. Now we're moving on to the renin-angiotensin-aldosterone system. So which of these is not true of angiotensinogen? So we know that we get renin released with low blood pressure, right? Renin will combine with angiotensinogen. Angiotensinogen is dormant, and it's always in the blood. So B would be true. Together, they travel to the liver, where the angiotensinogen converts to angiotensin 1. Okay, so it does combine with renin. It is always found in the blood. It does convert to angiotensin 1. Then the angiotensin 1 goes to the lungs, where ACE, angiotensin converting enzyme, is made, and that converts it from angiotensin 1 into angiotensin 2. Okay, and I'm gonna stop there and then we'll answer the next question. So let's go to, so 43, our answer was A. Does that make sense? It does not travel to the lungs. Good. Angiotensinogen does not travel to the lungs. It's always found in the blood. It does combine with renin. It does convert to angiotensin 1, but it does not travel to the lungs. It travels to the liver. Does that make sense? 44, aldosterone, which is not true of aldosterone. Good. So D is our correct answer, aldosterone. So what happens is angiotensin 2, let's change to green. Angiotensin 2 travels in the blood to the cortex of the adrenal gland, where aldosterone is produced, is made. Okay. When this arrival makes aldosterone be released, so aldosterone is then released, travels to the distal convoluted tubule and the collecting duct to cause reabsorption into the blood, of salt and water to raise blood pressure. So angiotensin 2 will travel to the cortex of the adrenal gland, causing the aldosterone to be released, which the aldosterone then goes to the distal convoluted tubule and collecting ducts, causing the blood to reabsorb salt, then water, which will then raise the blood pressure. So our answer would be D. Okay, aldosterone is not made in the medulla of the adrenal gland. It's made in the cortex of the adrenal gland. I know that one was a little bit tricky. You do want to go over these systems for your test. Kind of hard, but they are on there. 45, antidiuretic hormone. Okay, which of these is not true of antidiuretic hormone? Not true. I don't know what. I think I might have said true. I meant not true. Okay, maybe this is a tricky question. So we know that antidiuretic hormone is also called vasopressin, so A would be true. It is released from the posterior pituitary, B would be true. It does trigger the production of aquaporins, which basically cause a massive amount of water to backflow into the blood in order to raise blood pressure. So it is the only system that does not involve salt. It only installs aquaporins, and those aquaporins allow a massive amount of water to get back into the blood. So E would be correct because the antidiuretic hormone does not involve salt. Okay, so that one was a little bit maybe tricky. Okay, let's keep going here. Question 46. Alcohol serves as a natural diuretic to cause which of these? Good. E, right? So oliguria is just a little bit of urine. We know that if a diuretic makes you pee, right? If you drink alcohol, you have a few like beers or a glass of wine, you're peeing more. So it's serving as a natural diuretic, causing polyuria, which is excessive urination. So nephritis is like a diseased nephron. Cystitis is cysts in the bladder. Incontinence is not being able to hold your urine, right? Maybe you're bedwetting at night, or maybe you're an elderly person who, you know, has some type of neurological problem. You're incontinent. But alcohol is going to cause polyuria, so the answer there would be E. Okay, ammonia is excreted in urine as which of these compounds? Good. A, right? Urea has ammonia and carbon dioxide. So ammonia and carbon dioxide make up urea. Good. 48, secretion does not involve which of these? Okay, so remember we have our little glomerulus, our proximal convoluted tubule, our loop of Henle, our distal convoluted tubule, and our collecting duct. Okay. Step number 1, I'm going to try to just do this really quickly. Step number 1 is F for filtration. Filtration happens in the Bowman's capsule. Okay, step number 2 takes place in the loop of Henle and in the proximal convoluted tubule. Loop of Henle deals with water, proximal convoluted tubule deals with solutes, PCT, and loop, let's just do LH, are going to do number 2, which is R, reabsorption. Reabsorption. Does that make sense? And then last, number 3, secretion. Secretion happens in the distal convoluted tubule and the collecting duct. Remember, secretion is when the blood basically gives back the excessive solute that it accidentally, not accidentally, but it reabsorbed a lot of this solute in reabsorption. Remember, reabsorption, we take it back into the blood. Secretion, the body returns it out to be secreted in the urine. So S, or secretion, happens here. Okay, reabsorption happens here. Hopefully that visual kind of helps. This would be the blood here. Hopefully the visual helps you guys a little bit. So for question 48, secretion does not involve A, the proximal convoluted tubule, because the proximal convoluted tubule is not part of secretion. D is correct because the peritubular capillaries are looped around that distal convoluted tubule to facilitate, or they're performing the secretion. Okay, question 49, filtration. Which of these is not true about filtration? Good. So C, right? Blood cells are going to be too large to leave the fenestrae. Blood cells are too large, and so it only allows small particles or filtrate to leave the fenestrae, not blood cells. Blood cells are really big, so those are not going to fit through the fenestrae. Okay, question 50, and then I'm going to do like 10 more questions to lead us up to 45. So 50, reabsorption involves which of these? Good. B, the loop of Henle, right? 15 to 25% of reabsorption occurs in the loop of Henle. Okay, so hopefully, I know that those were kind of tricky questions. People wanted more questions, so I was trying to kind of find questions that hadn't been written before. So they are supposed to be, in terms of difficulty level, they're supposed to be the harder questions you would see on your exam. So to kind of end on a more uplifting note, let's spend the last 10 minutes. We're going to do some of the easier questions, the ones that should be faster for you guys, hopefully. And if you guys can, like before, kind of try to participate as much as you guys can in the chat so that I can see you guys answering. And we'll just do as many as we can in the 10 minutes we have left. I'm going to go to this right now. Which of these topics would you guys like to do? So I'm going to pull it up and then we can see. We've done muscle. We've covered all of these somewhat today. So say in the chat 1, 3, 5, or 7, because the other ones are answers. So do 1, 3, 5, or 7. Which one do you guys want to do right now? 1, 3, 5, or 7? Muscle, nervous, excretory, or reproductive? Okay, one more person to be our tiebreaker. Got 2 7s and 2 3s. Chris, I think you're our tiebreaker. 7 or 1, 3, 5. Well, you have to really say 7 or 3 now. Did we lose him? Maybe we lost him. I'm kind of leaning towards reproductive because we haven't done reproductive yet. Okay, so let's go for question 1. What is the dominant male hormone? Good. C, testosterone. 2, the male gonads are the E, right? The gonads themselves are the testes. The scrotum is the skin around the testes. The testes are the actual gonads. Does that make sense? Scrotum is the skin, testes are the gonads. 3, which muscle lines the walls of the scrotum? tricky. A little bit tricky. Yes, it's the dartus muscle. Dartus lines the entire wall. Cremaster lifts them. Maybe you could think of like a puppet master lifting. Cremaster lifts. Dartus, maybe you could think dart side to side. They line the walls. I'm just trying to help you remember it. So lines the walls, dartus. Maybe you could think like darting side to side. Cremaster, like a puppet master to lift them up. Okay, 4, which can undergo both meiosis and mitosis? D, right? Spermatogonia are the only cells that can undergo mitosis and meiosis. Good. Question 5, what is found inside the lobules? What is found inside the lobules? Good, the seminiferous tubules, right? The seminiferous tubules are found inside of the lobules. Good. 6, what is the storage place for sperm? Good, the epididymis. What is the storage place for sperm inside the testy? This is like a fill in the blank. Inside testy. What is the storage place for sperm inside the testy? Can anybody answer that? The storage place. The seminiferous tubule makes the sperm, but where do those seminiferous tubules collect or get together? Inside the testy. Outside the testy is the storage place, the epididymis. Inside the testy, there's one other. There we go, ret testy. So ret testy is the storage place for sperm inside the testy. Epididymis, storage place for sperm outside the testy. And then seminiferous tubule make the sperm themselves. Good. So the only reason they're not stored in the seminiferous tubule, they're traveling, but they don't actually stay there. Does that make sense? It's not a long-term storage place. 7, where is the nucleus of the sperm found? Good. D, or in the head of the sperm. Very good. 8, which does not belong? Good. D, ejaculatory duct. Good. 9, which enzyme does sperm secrete? Good. Acrosome. Very good. 10, which portion of the male urethra can be found outside of the abdominal region? Good. The spongy or cavernous urethra would be found outside the abdominal region. 11, what does the prostate add to the seminal fluid? remember that the prostate gland releases acid phosphatase and citric acid, both of which are enzymes that activate the tail, right? Fructose is the seminal vesicle. So seminal vesicle is for fructose. Acid phosphatate is the prostate plus citric acid. And the Cowper's or bulbourethral gland releases the alkaline mucus. Okay. Question 12. What is not part of the seminal fluid? This is kind of a weird question, actually. I'm thinking the answer is D. I didn't write these questions. I'm thinking the answer is D, but this is kind of an odd question. I'm going to go look for the answer real quick because that's kind of weird. I don't know exactly what they're wanting. Yeah, they're trying to say, so the answer is, oops. The answer is acrosome. They're trying to say, remember that the sperm is not part of the seminal fluid because the semen has seminal fluid and sperm. The sperm have acrosome. Does that make sense? The seminal fluid has everything besides sperm. So since the acrosome is a part of the sperm, it's not part of seminal fluid. Hopefully that made sense. Okay. Question, we're kind of narrowing to the end here. Let's see how many questions do we have? Whoa, okay, we can't get through all of these, but we're gonna keep going. Question 13, I'd like to finish male. Question 13, the foreskin is properly called. B, prepuce. 14, the removal of the prepuce. C, circumcision, good. 15, when the testes do not descend into the scrotum. Good. B, cryptorchidism. 16, let's do a few female. I know we have a few minutes. 16, which is a female sex hormone. Good. C, progesterone. 17, which does not belong. Good. A. 18. What is the term for the external female genitalia? Good. And remember, that's called vulva. External is vulva or pudendum. So it has two names. So don't miss pudendum. That could be on your test. 19. What is the oocyte surrounded by? So remember, it's surrounded by the follicle. The corpus luteum is the empty house with no oocyte. So the corpus luteum is the empty follicle. It has no oocyte. Corpus luteum is after ovulation. Corpus luteum after ovulation. Corpus luteum makes progesterone, and that basically sustains the uterine lining, but that's after the egg leaves. So the oocyte is always surrounded by a follicle. Okay, we only have 10 more. I think you guys are going quick. Why don't we just finish them? It's only going to take a few more minutes. 20. What is the longest portion of the fallopian tube? Longest portion of the fallopian tube. I actually don't know 100% what the answer is to this question. I think it should be A, which is what you guys are answering. I think it should be A, but just in case it's not, let's go make sure. I don't remember the answer to that. 20 is ampulla. Good. So remember, ampulla is where fertilization occurs. So question 20, A is the correct answer. The ampulla is where fertilization occurs. It's the longest part. The other two parts are the isthmus and the infundibulum. The isthmus enters into the uterus. The infundibulum is the first part. So it goes infundibulum, ampulla, isthmus. 21, what is the opening of the uterus? Good, the cervix, good. B, cervix. 22, which of the following holds the ovaries in place? Good. E, none. What is the correct answer? The correct answer there would be the ovarian ligament, right? And the broad, because the broad ligament kind of holds everything together. 23, which is not a part of the uterus? Which is not a part of the uterus? So I believe there is a myometrium, but you guys are answering A, so you're making me second guess myself. Let's go see. So there is a myometrium. So there's a peri, an endo, and a myometrium. You can kind of think of it or remember it as, you know, when a woman has a period, you need muscle to contract the blood out. You can also think of it as when you have a baby, you need the muscle of the uterus to contract to push the baby out. Does that make sense? So you have to have a muscle layer, myometrium. So where were we? Here, all of these would be part of the uterus. Myo, peri, endometrium, cervix, and fundus, all part of it, and the body of the uterus. So none would be correct because they're all part of the uterus. Question 24, where does fertilization normally take place? Good. D, fallopian tubes. Technically the ampulla of the fallopian tubes, but fallopian tubes would still be correct. 25, after ovulation, what does the follicle become? Good. C, corpus luteum. I just told you that, right? The corpus luteum is once we let the egg leave, it becomes the corpus luteum. 26, which layer of the uterus sloughs off during menstruation? Good. The endometrium, right? The myometrium is kind of pushing it out, but the endometrium is the one sloughing off. 27, which of the following is homologous to the glans? The glans penis. Good, the clitoris. 28, homologous to the scrotum. Good, the labia majora. That's homologous to the scrotum. You could think of it as the labia majora. Those are the fleshy filled outer. They grow pubic hair just like the scrotum grows pubic hair. 29, which is not part of the mammary glands. Kind of a weird question. Mammary glands are just breasts, and obviously breasts do not have skeletal muscle inside of them. You know, they have skeletal muscle at the base, but they don't have skeletal muscle inside of them. So it's kind of a weirdly worded question, but just know for your test, mammary gland is the same as breast. Last question, and then I'll let you guys go. So what causes the secondary sex characteristics in females? So that'd be like breast development, hair distribution, voice, all that. It is estrogen. So FSH is a follicle stimulating hormone. So follicle stimulating hormone helps the follicle to get bigger. Progesterone helps with pregnancy. You could think P, progesterone, pregnancy. It helps sustain the uterine lining. When progesterone falls, that's what causes a period. Estrogen is our correct answer. Estrogen is what basically gives the development of the follicle, helps with preparing the female body to be female with all of the different secondary sex characteristics. Estrogen develops the follicle along with FSH, and then progesterone is going to be what sustains the uterine lining. LH is luteinizing hormone, or D is released with ovulation. So the answer to this question is B. Okay, so I will see you guys on Thursday. I feel like the practice questions are good. I think I'm going to do more practice questions with you guys if you guys like. Hopefully you see how the difficulty level of the questions can kind of help root out where your guys' weak areas are. Just kind of judging off your guys' answers, what I would recommend, you know, from kind of where I saw you guys missing questions, seems like muscle. You guys need some review in the muscle area. Seems like muscle is an area where you guys should spend some time reviewing. And then it seems like maybe kidney would be good to review too. But I will see you guys Thursday morning. Thank you guys for staying. And we'll do more practice questions then. And let me know if you guys have questions on Canvas.